Good morning, everyone. I am pleased to welcome you all today. Thank you for joining this session. Today, we will be discussing the incorporation of recovery processes in sterile dosage forms and its impact on shelf life. This presentation will explore how recovery processes are integrated into sterile formulations and what effects they have on the stability and shelf life of the products. Let's get started and delve into this important topic together. Welcome to the introduction. Today, we'll be focusing on sterile dosage forms, such as injections, ophthalmics, and infusions. These types of products require absolute sterility and a high level of stability to ensure safety and efficacy. Because they are so sensitive, they are highly vulnerable to microbial contamination, which can pose serious health risks. They are also at risk from particulate matter, which can cause blockages or adverse reactions, and chemical degradation, which can compromise the product's effectiveness. It's important to understand that the recovery processes we use to manufacture and handle these products can introduce significant regulatory and quality risks. Ensuring the sterility and stability of these forms is a critical part of our overall quality assurance efforts, and we'll explore how to manage these challenges effectively as we go along. Let's talk about recovery processes. These are methods used when something doesn't go as planned during sterilization or sterilization-related steps. For example, reprocessing might involve refiltration or re-sterilization to ensure the product is safe and effective. Reworking could mean adding an extra sterile processing step to address any issues identified. Sometimes, if filtration fails, we need to recover the product to prevent waste and ensure patient safety. Now, from a regulatory perspective, these recovery processes are generally discouraged or even prohibited unless there's a very solid justification. They require extensive validation and thorough risk assessments to prove that they don't compromise safety or quality. Essentially, any recovery process must be carefully justified and validated to meet compliance standards. Connecting this to the overall theme, understanding these processes helps us appreciate the importance of maintaining strict control and validation in sterile processing, because patient safety depends on it. Let's take a closer look at the risk mechanisms of recovery and how they can impact product quality. First, refiltration is a common process, but it carries a contamination risk if not done properly. Next, re-sterilization is necessary at times, but it can lead to thermal degradation of the product, potentially affecting its efficacy. Additional exposure during handling or storage can cause oxidation or increase light sensitivity, which may compromise the stability of the product. Reopening the container also poses a risk of breaching sterility, which can introduce contaminants. All of these factors ultimately lead to a reduced shelf life or even batch rejection, highlighting the importance of strict control measures. Now, when we talk about critical quality attributes, or CQAs, for sterile products, these are the key parameters we monitor to ensure safety and efficacy. For example, the sterility assurance level, or SAL, is a critical measure of how confidently we can say a product is free of viable microorganisms. Endotoxin levels are equally important, as they can cause severe reactions if present in contaminated products. We also look at assay results, such as potency percentages, to confirm the product's effectiveness. Degradation products, particulate matter, and pH levels are other vital CQAs, all contributing to the overall quality profile. Most importantly, any deviation from these attributes can directly impact patient safety. That's why rigorous control and monitoring are essential at every stage of the process to ensure we deliver safe, effective sterile products. Let's talk about some of the stability concerns we face with sterile dosage forms. First, chemical instability. This includes processes like hydrolysis and oxidation, which can degrade the drug's effectiveness over time. It's crucial to understand these because they can lead to reduced potency or even formation of harmful byproducts. Next, we have physical instability. This involves issues like precipitation and aggregation, which can affect the drug's appearance and potentially its safety or efficacy. These physical changes are often visible and can be a sign that the formulation is deteriorating. Then there's the microbiological risk. Maintaining sterility is vital, but it can be compromised during various stages, especially during recovery steps. When we recover or prepare the product, there's a risk of losing that sterile barrier, which could lead to contamination. And that's a key point. Recovery steps, while necessary, can sometimes jeopardize these stability attributes. So, it's a constant balancing act to ensure we keep the drug stable and sterile throughout its life cycle, especially during handling and processing. All right, let's walk through this calculation example. Before recovery, we have a rate constant, K, of 0.001 month to the power of -1. Using that, we calculate the time to 90% sterilization, or T90, by dividing 0.105 by the K value, which gives us 105 months. This means, initially, the product has a fairly long shelf life. Now, after re-sterilization, the rate constant increases to 0.006 month to the -1. When we do the same calculation, 0.105 divided by 0.006, we get approximately 17.5 months. That's a significant decrease in shelf life. The key takeaway here is that heat exposure during re-sterilization markedly accelerates the sterilization process, drastically reducing the product's shelf life. This example highlights why maintaining optimal sterilization conditions is so crucial for preserving product stability over time. Let's take a closer look at endotoxin risk, using this example to illustrate the key points. According to the USB, the limit for endotoxins in typical IV products is 5 EU per kilogram of body weight. Now, before recovery, the endotoxin level might be around 0.2 EU per milliliter, which is well below the limit. However, after recovery, this level can increase to about 0.8 EU per milliliter. This increase is often due to handling, which can introduce additional endotoxins into the product. The concern here is that such an increase could elevate the risk of a pyrogenic reaction, something we definitely want to avoid. So, the main takeaway is that handling can impact endotoxin levels, and even small increases can pose health risks. It's crucial to monitor and control these levels throughout the process to ensure patient safety and comply with regulatory standards. This example underscores the importance of strict procedures and quality control in managing endotoxin risks effectively. Let's take a closer look at the stability study design. According to ICH guidelines, we have different conditions for long-term and accelerated testing. For long-term stability, samples are stored at either 25 degrees C with 60% relative humidity or at refrigerated conditions of 2 to 8 degrees Celsius. These conditions help us understand how the product behaves over time under typical storage scenarios. Then, we also conduct accelerated stability testing at 40 degrees C with 75% relative humidity. This helps us predict the product's shelf life by subjecting it to harsher conditions to see how it might degrade faster. Now, regarding the test parameters, we focus on several critical quality attributes. Sterility is essential to ensure the product remains free from microbial contamination. Endotoxins testing helps confirm that bacterial toxins are not present at harmful levels. The assay, of course, verifies the potency and content of the active ingredient. Particulates are also monitored to prevent any foreign particles from being present, which could pose safety risks. And an important aspect is comparing the recovered product after storage or stress conditions to the original batch to see if there are any significant changes or losses in quality. This comprehensive approach ensures that the product remains safe, effective, and consistent throughout its shelf life, aligning with our overall goal of delivering reliable pharmaceutical products. Let's consider a hypothetical case study to bring these concepts to life. Imagine we have a product, a sterile injectable antibiotic. Now, this example will help us understand how the principles we've discussed apply in real-world scenarios. When developing or reviewing such a product, key points include ensuring strict sterility, proper formulation, and rigorous quality control measures. These factors are crucial because they directly impact patient safety and treatment efficacy. By analyzing this case, we can see how adherence to regulatory standards and best practices ensures the product's success from development through to the end user. This example ties back to our overall theme of quality assurance and compliance in pharmaceutical manufacturing. It underscores the importance of a comprehensive approach to each step in the process, especially for sensitive products like injectables. Let's talk about the regulatory expectations surrounding reprocessing in sterile products. It's quite strict. Reprocessing is highly restricted because the risk of compromising sterility is significant. When it is allowed, it must be fully validated to ensure safety and efficacy. Reprocessing is only permissible if certain conditions are met. First, sterility assurance must be maintained throughout the process. This means that the product remains sterile and safe for use. Second, there should be no increase in endotoxins or particles, as these could pose serious health risks. And third, the stability of the product must remain unchanged after reprocessing. If these conditions are not met, the consequences are clear. Batch rejection is mandatory. That's a strict but necessary measure to ensure patient safety and product integrity. Overall, these regulations emphasize that reprocessing isn't taken lightly. It's only allowed under very specific, controlled circumstances to ensure the highest standards of safety and quality in sterile products. Let's wrap up with some key takeaways. Sterile dosage forms, as we've discussed, demand the highest quality standards to ensure patient safety and product efficacy. When we introduce recovery processes, there are important risks to consider. These include the potential for sterility breaches and increased degradation of the product. It's crucial that shelf life is not just estimated arbitrarily, but is scientifically justified and supported by thorough stability data. This ensures that the product remains safe and effective throughout its intended shelf life. Remember, recovery in sterile products can be acceptable only if there's strict regulatory compliance. This compliance must demonstrate clearly that the recovery process does not compromise sterility, safety, or the product's shelf life. Ultimately, maintaining these standards safeguards both the integrity of the product and the trust of those who rely on it. For further support, please contact us. We're here to help you with any questions or additional information you might need. Just reach out, and we'll be happy to assist. Thank you so much for your time and attention today. We appreciate your engagement and look forward to supporting you moving forward.